Haplogroup R1b is the dominant paternal lineage of Western Europe. In human genetics, haplogroup R1b is the most frequently occurring Y-chromosome haplogroup in Western Europe and in parts of sub-Saharan Central Africa. R1b is also present at lower frequencies throughout Eastern Europe, Western Asia, Central Asia, and parts of North Africa, South Asia, and Siberia. Due to European emigration, it also reaches high frequencies in the Americas and Australia. While Western Europe is dominated by the R1b1a2 branch of R1b, the mostly Chadic-speaking area in Africa is dominated by the branch known as R1b1c. These represent two very successful twigs on a much bigger family tree. Nomenclature, R1b, R1b1, and so on are phylogenetic, or family tree-based names which explain the branching of the family tree of R1b. For example, R1b1a and R1b1b would be branches of R1b1, descending from a common ancestor. This means that these names can change with new discoveries. The alternative way of naming haplogroups is to refer to the SNP mutations used to define and identify them, for example RM343, which is equivalent to R1b. Haplogroup R1b is in other words now identified by the presence of the single nucleotide polymorphism mutation M343, which was discovered in 2004. From 2002 to 2005, R1b was defined by the presence of the SNP named P25. Standardized naming as described above, both using phylogenetic or mutational systems, was first proposed in 2002 by the Y-chromosome consortium. Prior to 2002, today's haplogroup R1b had a number of names in differing nomenclatural systems, such as HG1 and U18. After 2002, a major update of the YCC phylogenetic nomenclature was made in 2008 by Karafit al., which took account of newer discoveries of branches which could be clearly defined by SNP mutations, including some which changed the understanding of R1b's family tree. Since 2008 it has become increasingly necessary to refer to the frequently updated listing made on the ISOGG website. Also prior to 2002, major Y-DNA signatures based on markers other than SNPs were recognized. In Western Europe, the STR haplotype known as the Atlantic modal haplotype was found to be most common by Wilson al. Even earlier research using RFLP genotyping identified two distinct haplotypes within RM269. In Southeast Europe and Southwest Asia, haplotype 35, or HT35 was found to be a common form of RM269, whereas in Western Europe haplotype 15, or HT15 dominated in frequency. Origin and dispersal. R1b is a subclade within the much larger Eurasian MNOPS macrohaplogroup, which is one of the predominant groupings of all the rest of human male lines outside of Africa, and this whole group, along indeed with all of macrohaplogroup F, is believed to have originated in Asia. The point of origin of R1b is thought to lie in Eurasia, most likely in Western Asia. T. Karafit al. estimated the age of R1, the parent of R1b, as 18,500 years before present. Early research focused upon Europe. In 2000 Ornella Semino and colleagues argued that R1b had been in Europe before the end of the Ice Age, and had spread north from an Iberian refuge after the last glacial maximum. Age estimates of R1b in Europe have steadily decreased in more recent studies, at least concerning the majority of R1b, with more recent studies suggesting a Neolithic age or younger. Only Morley al. have recently attempted to defend a Paleolithic origin for R1b1b2. Irrespective of STR coalescence calculations, Chaiki al. pointed out that the timing of molecular divergences does not coincide with population splits. The TMRCA of haplogroup R1b dates to its point of origin somewhere in Eurasia, and not its arrival in Western Europe. 1. However, Michael Amalio argues that the closest branch of R1b is from Iberia and its small subclades found in West Asia. The Near East and Africa are examples of back migration, and not of its origin. Barbara Arady and colleagues were the first to point out that the distribution of R1b STR variants in Europe forms a cline from east to west, which is more consistent with an entry into Europe from Western Asia with the spread of farming. a 2009 paper by Cameroon et al. Added to this perspective by using R1b as an example of a wave haplogroup distribution, in this case from east to west. The proposal of a southeastern origin of R1b was supported by 3 detailed studies based on large data sets published in 2010. These detected that the earliest subclades of R1b are found in Western Asia and the most recent in Western Europe. While age estimates in these articles are all more recent than the last glacial maximum, all mention the Neolithic, when farming was introduced to Europe from the Middle East as a possible candidate period. Myers et al. And Cruciani et al. Both remained undecided on the exact dating of the migration or migrations responsible for this distribution, not ruling out migrations as early as the Mesolithic or as late as Hallstatt but more probably late Neolithic. They noted that direct evidence from ancient DNA may be needed to resolve these gene flows. Lee et al. Analyzed the ancient DNA of human remains from the late Neolithic Belbeker site of Kromsdorf, Germany, identifying 2 males as belonging to the Y haplogroup R1b. Analysis of ancient Y DNA from the remains of populations derived from early Neolithic settlements such as the Mediterranean Cardium and Central and North European LBK settlements have found an absence of males belonging to haplogroup R1b. It has thus been suggested that the arrival of Y haplogroup R1b coincided with the arrival of the Centum branch of the Indo-European languages in Western Europe. In Blood of the Isles, Brian Sykes gives the populations associated with haplogroup R1b the name Oiza for a clan patriarch, much as he did for mitochondrial haplogroups in his work The 7 Daughters of Eve. European R1b is now known to be dominated by RM269, and the origins of this branch are discussed further in more detail below. Root of R1b tree, for clarity, the identifiers below are those from both the 2010 and 2011 revisions of the ISOGG tree. R1b is extremely rare. The only population yet recorded with a definite significant proportion of R1b are the Kurds of southeastern Kazakhstan with 13%. However, more recently, a large study of Y-chromosome variation in Iran revealed R1b as high as 4.3% among Persian subpopulations. In a study of Jordan, it was found that no less than 20 out of all 146 men tested, including most notably 20 out of 45 men tested from the Dead Sea area, were positive for M173 but negative for P25 and M269, mentioned above, as well as the R1 markers SRY 10831.2 and M17. A study indicates that they are all R1b2V88, 2. Hassan et al. Found an equally surprising 14 out of 26 of Sudanese Fulani who were M173 plus and P25. Wood et al. Report 2 Egyptian cases of R1 M173 which were negative for SRY 10831 and P25, out of a sample of 1,122 males from various African countries, including 92 from Egypt. Such cases could possibly be either R1b or R1a. It is however also possible that some of the rare examples represent a reversion of marker P25 from a positive back to a negative ancestral state. Frequency table of R1b1 Subclades. An up-to-date compilation of data taking the latest information into account can be found in Cruciani et al., which can be summarized as follows. As will be discussed below, however, in some parts of Western and Northwestern Europe, RM269 frequencies can reach even higher levels. R1b1, like R1b, is rare. As mentioned above, examples are described in older articles, for example 2 in a sample from Turkey, but most cases, especially in Africa, are now thought to be mostly in the more recently discovered subclade V88. Most or all examples of R1b therefore fall into subclades R1b1c or R1b1a. Cruciani et al. In the large 2010 study found 3 cases amongst 1173 Italians, 1 out of 328 West Asians and 1 out of 156 East Asians. Vazari found 3 cases in the Ukraine, in a study of 322 people from the Dniester-Carpathian region, who were P25 positive, but M269 negative. Cases from older studies are mainly from Africa, the Middle East or Mediterranean, and are discussed below as probable cases of R1b1c. R1b1a, R1b1a is defined by the presence of SNP marker P297. In 2008 this polymorphism was recognized to combine M73 and M269 into one R1b1a cluster. The majority of Eurasian R1b is within this clade, representing a very large modern population. Although P297 itself has not yet been much tested for, the same population has been relatively well studied in terms of other markers. Therefore the branching within this clade can be explained in relatively high detail below. R1b1a1, poor SNP typing has caused confusion as to whether this group is a 1b or Q. For example, in Bihor Al5 former cases of R1bm73 were relabeled as a subclade of PQR2XR1, suggesting that this group may really be Q or less likely R2, as the so-called R1bm73 samples were negative for the mutation that defined R1. M73 is defined by the presence of SNP marker M73. It has been found at generally low frequencies throughout Central Eurasia, and in Altai, but has been found with relatively high frequency among particular populations there including Hazaras in Pakistan and Bashkirs in Bashkortostan, 44 of these being found among the 80 tested Bashkirs of the Upselilovsky district in the Republic of Bashkortostan. 4 M73 men were also found in a 523-person study of Turkey, and 1 person in a 168-person study of Crete. A large proportion of Commandin Y chromosomes belong to M73, which is largely restricted to Central Asia but has also been found in Altayan Kazakhs and other southern Siberians. In 2007, Myers al. report that out of 193 M73 men found amongst 10,355 widespread men, all except 2 Russians occurred outside Europe either in the Caucasus, Turkey, the Circum-Uralic and North Pakistan regions. R1b1a2, R1b1a2 is defined by the presence of SNP marker M269. R1b1a2 or M269, XL23, is found at highest frequency in the Central Balkans, notably Kosovo with 7.9%, Macedonia 5.1% and Serbia 4.4%. Kosovo is notable in also having a high percentage of descendant L23 or L23, XM412. At 11.4% unlike most other areas with significant percentages of M269 and L23 except for Poland with 2.4% and 9.5% and the Bashkirs of southeast Bashkortostan with 2.4% and 32.2% respectively. Notably this Bashkir population also has a high percentage of M269 sister branch M73 at 23.4%. 5 individuals out of 110 tested in the Ararat Valley, Armenia belong to R1b1a2 and 36 to L23, with none belonging to known subclades of L23. European R1b is dominated by RM269. It has been found at generally low frequencies throughout Central Eurasia, but with relatively high frequency among Bashkirs of the Perm region and Bamyk-Sky region. This marker is also present in China and India at frequencies of less than 1%. The table below lists in more detail the frequencies of M269 in various regions in Asia, Europe, and Africa. The frequency is about 82% in Ireland, 70% in Scotland, 68% in Spain, 60% in France, 45% in Eastern England, 50% in Germany, 50% in the Netherlands, 42% in Iceland, and 43% in Denmark. It is as high as 95% in parts of Ireland. It is also found in some areas of North Africa, where its frequency peaks at 10% in some parts of Algeria. From 2003 to 2005 what is now R1b1a2 was designated R1b3. From 2005 to 2008 it was R1b1c. From 2008 to 2011 it was R1b1b2. As discussed above, in articles published around 2000 it was proposed that this clade had been in Europe before the last ice age, but by 2010 more recent periods such as the European Neolithic have become the focus of proposals. A range of newer estimates for R1b1b2, or at least its dominant parts in Europe, are from 4,000 to a maximum of about 10,000 years ago. and looking in more detail is seen as suggesting a migration from Western Asia via southeastern Europe. Western European R1b is dominated by P310. It was also in this period between 2000 and 2010 that it became clear that especially Western European R1b is dominated by specific subclades of RM269. Within Europe, RM269 is dominated by RM412, also known as RL51, which according to Myers al. Is virtually absent in the Near East, the Caucasus and West Asia. This Western European population is further divided between a P312 per second 116 and a U106 per second 21, which appear to spread from the western and eastern Rhine River basin respectively. Myers al. Note further that concerning its closest relatives, in L23, that it is instructive that these are often more than 10% of the population in the Caucasus, Turkey, and some Southeast European and Circum-Uralic populations. In Western Europe it is also present but in generally much lower levels apart from an instance of 27% in Switzerland's Upper Rhone Valley. In addition, the subclade distribution map, Figure 1a titled L11, XU106, S116, in Myers al. shows that a P310 per litre 11 occurs only in frequencies greater than 10% in central England with surrounding areas of England and Wales having lower frequencies. This P310 per litre 11 is almost non-existent in the rest of Eurasia and North Africa with the exception of coastal lands fringing the western and southern Baltic and in eastern Switzerland and surrounds. In 2009, DNA extracted from the femur bones of 6 skeletons in an early medieval burial place in Igolding dated to around 670 AD yielded the following results. 4 were found to be haplogroup R1b with the closest matches in modern populations of Germany, Ireland and the USA while 2 were in haplogroup G2a. Population studies which test for M269 have become more common in recent years, while in earlier studies men in this haplogroup are only visible in the data by extrapolation of what is likely. The following gives a summary of most of the studies which specifically tested for M269, showing its distribution in Europe, North Africa, the Middle East and Central Asia as far as China and Nepal. R1b1a2a1a1a, this subclade is defined by the presence of the SNPU106, also known as S21 and M405. It appears to represent over 25% of R1b in Europe. While this subclade of R1b is frequently discussed amongst genetic genealogists, the following table represents the peer-reviewed findings published so far in the 2007 articles of Myers al. and Sims al. R1b1a2a1a2, along with RU106, RP312 is one of the most common types of R1b1a2 in Europe. Also known as S116, it has been the subject of significant study concerning its subclades, and some of the ones recognized by the ISOGG website are summarized in the following table. Myers al. described it distributing from the west of the Rhine basin. Amongst these, scientific publications have given interpretation and comment on several. RP312-2 unites several branches of RP312. RP312-2A is defined by the presence of the marker M153. It has been found mostly in Basques and Gascons, among whom it represents a sizable fraction of the Y-DNA pool, though is also found occasionally among Iberians in general. The first time it was located it was described as H102 and included 7 Basques and 1 Andalusian. RP312-2b. This subclade is defined by the presence of the marker L176.2. It contains the following. RP312-2 bar. This subclade is defined by the presence of the marker M167, also known as SLY2627. The first author to test for this marker was Hels in 1999, who tested 1158 men in various populations. He found it relatively common among Basques and Catalans. Other occurrences were found among other French, British, Spaniards, bar copyright ANAS, and Germans. In 2000 Rossel al., in a study which tested 3616 men in various populations also tested for that same marker, naming the haplogroup HG22, and again it was found mainly among Basques, in lower frequencies among French, Bavarians, Spaniards, Southern Portuguese, and in single occurrences among Romanians, Slovenians, Dutch, Belgians and English. In 2001 Bosch described this marker as H103, in 5 Basques and 5 Catalans. Further regional studies have located it in significant amounts in Asturias, Cantabria and Galicia, as well as again among Basques. Cases in the Azores have also been reported. In 2008 2 research papers by Larcubed Pez-Para and Adams, respectively, confirmed a strong association with all or most of the Pyrenees and eastern Iberia. In a larger study of Portugal in 2006, with 657 men tested, Beliza et al. Confirmed similar low levels in all the major regions, from 1.5%-3.5%. RP312-2b2. This subclade is defined by the presence of the marker S68, also known as L165. It is found in England, Scandinavia, and Scotland. It has been suggested, therefore, that it arrived in the British Isles with Vikings. RP312-3 is defined by the presence of the marker U152, also called S28. Its discovery was announced in 2005 by Ethno-Ancestry and subsequently identified independently by Sims et al. Myers et al. Report this clade is most frequent in Switzerland, Italy, France and western Poland, with additional instances exceeding 15% in some regions of England and Germany. Similarly Cruciani et al. Reported frequency peaks in northern Italy and France. Out of a sample of 135 men in Tyrol, Austria, 9 tested positive for U152 per second 28. Far removed from this apparent core area, Myers et al. Also mention a subpopulation in North Bashkortostan where 71% of 70 men tested were in U152. They propose this to be the result of an isolated founder effect. RP312-4 is defined by the presence of the marker L21, also referred to as M529 and S145. Myers et al. Report it is most common in England and Ireland. Known subclades include the following: RP312-4b. This subclade within L21 is defined by the presence of the marker M222. It is particularly associated with male lines which are Irish or Scottish, but especially Northern Irish. In this case, the relatively high frequency of this specific subclade among the population of certain counties in northwestern Ireland may be due to positive social selection, as it is suggested to have been the Y-chromosome haplogroup of the Waynar copyrightal dynastic kindred of ancient Ireland. However, it is not restricted to the Waynar copyrightal as it is also associated with the closely related Canotta dynasties. The Way-Brian and Way-Fiacrach M222 is also found as a substantial proportion of the population of Scotland, which may indicate substantial settlement from Northern Ireland or at least links to it. Those areas settled by large numbers of Irish and Scottish emigrants such as North America have a substantial percentage of M222. RP312-4F. This subclade within L21 is defined by the presence of the marker L159 and is known as L159.2 because of a parallel mutation that also exists inside haplogroup I2A1. L159.2 appears to be associated with the kings of Leinster and Diamate Mac Mercada. It can also be found in the coastal areas of the Irish Sea including the Isle of Man and the Hebrides, as well as Norway, western and southern Scotland, northern and southern England, northwest France, and northern Denmark. RP312-4G. This subclade within L21 is defined by the presence of the marker L193. Many surnames with this marker are associated geographically with the western border region of Scotland. A few other surnames have a Highland association. RL193 is a relatively young subclade likely born within the last 2,000 years. RP312-4 hours. This subclade within L21 is defined by the presence of the marker L226, also known as S168. Commonly referred to as Irish Type 3, it is concentrated in central western Ireland and associated with the Dar-LGCAIS kindred. RDF21. This subclade within L21 is defined by the presence of the marker DF21 also known as S192. It makes up about 10% of all L21 men and is c. 3,000 years old. R1b1b. R1b1b is defined by the presence of SNP marker M335. This haplogroup was created by the 2008 reorganization of nomenclature and should not be confused with R1b1b2, which was previously called R1b1c. Its position in relation to the much more popular subclade R1b1a is uncertain. The M335 marker was first published in 2004, when one example was discovered in Turkey, which was classified at the time as R1b4. R1b1c. R1b1c is defined by the presence of SNP marker V88, the discovery of which was announced in 2010 by Cruciani et al. The V88 group in Africa may be linked with Chadic languages and an Iberian origin. Apart from individuals in southern Europe and western Asia, the majority of our V88 was found in northern and central Africa, as can be seen in the above data table. R1b1c is found in northern Cameroon in west-central Africa at a very high frequency, where it is considered to be caused by a pre-Islamic movement of people from Eurasia. Suggestive results from other studies which did not test for the full range of new markers discovered by Cruciani et al. Have also been reported, which might be in a V88. Wood et al. Reported high frequencies of men who were P25 positive and M269 negative, amongst the same North Cameroon area where Cruciani et al. Reported high RV88 levels. However, they also found such cases amongst 3% of Fanti from Ghana, 9% of Bassa from southern Cameroon, 4% of Herero from Namibia, 5% of Ambo from Namibia, 4% of Egyptians, and 4% of Tunisians. Lewis et al. Found the following cases of men M173 positive, but negative for M73, M269, M18 and M17, 1 of 121 Omanis, 3 of 147 Egyptians, 2 of 14 Bantu from southern Cameroon, and 1 of 69 Hutu from Rwanda. Pereira et al. In a study of several Saharan Tuareg populations, found 1/3 of 31 men tested from near Tanut in Niger to be an R1b. R1b1c1. R1b1c1 is a subclade of RV88 which is defined by the presence of SNP marker M18. It has been found only at low frequencies in samples from Sardinia and Lebanon. M73, Y-chromosomes known as R1b1m73 first appeared in the literature of Sengupta et al. In 2006 among 5 out of 25 Hazaras. R1b1m73 was the sole type of R1b found in the Klimandin and a dominant clade in the southeastern Bashkirs. However, a recent 2010 study by Bihor et al. Showed that these same 5 Hazara did not belong to R1b but to A2 or Q. If this study by Bihor is right, then the Klimandins have no R1b. Ironically, the Bashkirs still have a high percent of R1b even though some of the southern Bashkirs had mislabeled Q which appeared to be R1b due to confusion between Q1AM25 and R1bp25. If Bihor et al. Is right about M73, then the southeastern Bashkirs have both R1b and Q while all the other Bashkirs have a high frequency of R1b while only having 1 to 2% of Q. Historical note: the DNA tests that assisted in the identification of Tsar Nicholas II of Russia found that he had haplogroup R1b. In popular culture, Brian Sykes, in his book Blood of the Isles, gives the populations associated with R1b the name of Oiza N for a clan patriarch, much as he did for mitochondrial haplogroups in the 7 Daughters of Eve. Stephen Oppenheimer also deals with this haplogroup in his book Origins of the British, giving the R1b clan patriarch the Basque name Ruisco in honor of what he thinks is the Iberian origin of R1b. Artem Lukikov has created an animation based on a Bashkir epic about the Ural, which outlined the history of the clusters of haplogroup R1, R1a and R1b. See also, human Y-chromosome DNA haplogroup, Atlantic modal haplotype, genealogical DNA test, prehistoric Europe, Y-DNA haplogroups in European populations. References